This is a genome. It represents one microorganism and gives it the instructions to grow and reproduce. If you notice, this genome is circular. Bacterial and archaeal genomes are circular, unlike human genomes, which are linear and bunch up to form the chromosomes that you've probably seen before. If I want to sequence this genome, that is, figure out the specific order of A's and T's and C's and G's, I can do that relatively easily so long as I have a pure culture. That is, just one genome with lots of copies. All I would have to do is isolate this DNA from the rest of the cell and use one of many commercially available sequencing methods that I won't go into in this video. Genome sequencing will give us the DNA sequence back in fragments. We put these fragments through a computer algorithm to match the overlapping sequences of DNA, reforming the genome digitally. If the overlapping sequences form a circle, we have what is called a closed genome, and we know with relative certainty that this is the complete genome for this organism. Otherwise, we would have a linear, open genome that contains more information that we don't know. But now, say that we start with an environmental sample, not a pure culture. This process gets a lot more difficult. We still have to isolate the DNA and sequence it the same way, but now we have a bunch of different fragments from a bunch of different organisms, and we need to figure out where they all came from. So we developed algorithms that use lots of computational power to try and assemble these fragments back into the genomes they came from. It tries a bunch of different assemblies and outputs the best genomes it could make, fittingly called metagenome assembled genomes, or MAGs. MAGs are usually worse quality than genomes that come from pure cultures, but metagenomes can give us more information about the community. For example, if we get 5 genomes from one organism and only 1 genome from another, we can say that the relative abundance of organism 1 to organism 2 is 5 to 1. This is just one of many extra pieces of data that metagenomes can provide.